Good afternoon, everyone. We'll go ahead and get started. We have a few people across the hall at a gaming meeting, and Tom is walking his dog. So thank you all for coming. Look forward to seeing all the presentations, and I'll turn it over to Matea for her part of this. Thank you.
All right, you can hear me okay? Okay, so our first presentation is Dr. Anissa Goforth and Dr. Jingjing Sun. So I will call them up.
Good afternoon. My name is Anissa Goforth. My colleague Jingjing Sun is unable to be here. She's visiting her family in China. I'm so grateful to Tribal Council to have this opportunity to speak with you, to share the work that we've been doing for the last 8 years, as well as some new adventures we are having together in Ashili, and as well from Arlie. I want to start thinking about, I'm coming from the University of Montana, and one of the things that I came into my profession as a school psychologist was that research was this thing that we do to examine children. So I focus on kids. And over the years, over my 15 years, I've come to see research as a relationship. That the work that I do as a researcher, as a psychologist, as someone who's interested in supporting children's mental health, is that research is with community. It is with community in a way that is driven by the ideas of those whose strengths, who know their community best. And as an outsider, as a visitor to here in a Flathead Nation, that's how Jingjing and I have come to approach our research. We are here as a resource from the University of Montana. We are not coming in as experts. We are not coming in as anything other than someone to walk alongside the communities that we've gotten to know, and very honored to be here with several of the community advisory members who've been here together. About 8 years ago, the superintendent at Arlie School District had reached out to us and said, our children need support. And will you help us think about a way to make a social emotional learning program that is culturally based, that is land-based, that is connected to their culture and identity. And as someone myself, that's the research that I do. I'm really interested in that children should connect their identity with their schooling. And over the last 5 years, I've had the privilege of working with a community advisory board, educators, staff, in developing what we have come to call pathways to wellness. And on the screen in front of you, you'll see the pathways to wellness, and Rhonda Howlett here is going to talk a little bit more in a moment. But that image also next to it is the picture of us in front of 9 pipes. These are many of the community members that have been involved in this project, where we developed pathways to wellness to really think about how we can develop a program that is in public schools, that is connected with who they are, and social emotional learning, meaning how we can understand emotional regulation, how we can develop peer relationships as kids, how we can support their overall mental health. About 2 years ago, I think it was, the Salish Culture Committee, we had presented in front of them, and the elders said, this is wonderful, we really support it, but you're only in Arlie. Can you think about maybe expanding to a different community? The University of Montana team, we found a grant from the Robert Wood Johnson Foundation and said, yes, we can apply for this grant, and we can think about working with Michelle Mitchell in tribal education and think about what communities might benefit from some additional support. And over consultation, Michelle Mitchell reached out to Councilwoman Danielle Matt and others to think that Eschalip might be a really wonderful community to connect with. And over the past year, we've met with community members and parents and teachers and students to understand what we can do in Eschalip that might be different or similar to what we've done in Arlee. Our goal as researchers isn't to get some program in New York City or California and say, okay, we're going to plop this in Arlee and say, hey, this is going to work, right? No, we knew that immediately that had to come from the people who are part of the community. And so for this next stage, and we have Sarah Big Sam going to talk a little bit about what we've been doing, is thinking about, do we take something that was developed from Arlee and just copy it into Eschalip? When, as you can see from the poster there in the bigot in the middle, that's very place-based, that's very focused on Arlee. What does it look like though, maybe in Eschalip? And that's our next step in our journey for the next 2 years, is thinking about what this can look like for the school, what this look like for the community, and our eagerness and humbleness to work with the community members who are really committed to their children. And on that note, I will pass it over to Rhonda Howlett, who has been involved right from the beginning.
Good afternoon, and it's my pleasure to be here in Pueblo, talking to you guys and understanding that we're all for the kids, right? And we've been doing this project with these ladies from U of M, like she said, for about 8 years. But what I understand is that, there's so much, history on our reservation with mistrust with researchers. And, I'm here also to assure you that the relationship that we've built with this team from U of M is just one of the most high level, integrity-based, relationship-based program that I've ever worked with. And knowing that a lot of times when we have grants, they come in and do their thing, and then they're out the door, and we never see them again. But this isn't the case. When I started with the CAB, which is the Community Advisory Board in Arlee, I also joined with several other community members, but there was like Adele Martin and Patty Stevens and Julie Kajun and Debbie Hoganson, and those are just a few, but we had a really good representation of our community, including the school psychologist and the school counselor. And Debbie here on my right is a retired educator, and I would say that because of the timing of this grant, it was like a perfect storm for us to be involved and to have something really meaningful to work on as retired people that are in the from the classroom and just have our heart with kids. And so I know that this is from the class for us to be able to work with them and to and my feeling is that I've always really been my degree in education went up to the master's level, and it's in curriculum and instruction. And so I know that gave me the pathway to really use that education. Debbie here is, she worked in Cherry Valley School, I believe, and then she worked down with her husband down in Las Vegas, and then came back and retired out of Arlee School District. And so she was very familiar with our kids too, and followed them with their sports and different things. And so that time that we worked together, initially trying to figure out what it was with our community and social emotional learning, and how we could perceive it being part of who we are. And Debbie is just really into the language and vocabulary of education, and she's a reading specialist. And so her second time that we ever met during this CAB meeting time, she said, when are we going to start writing lessons? And it was like, okay, well, let's get on it. So we looked at the national model that they had, and it didn't really have curriculum attached to it, but it had a framework that we could go from. And that framework that we worked from, excuse me, had relationship skills, self-management, social awareness, self-awareness, and responsible decision-making. And so those were the national level social emotional learning goals. And with me, you know, those were ones that I thought, well, we can work with that. But what happened then is that we, you know, with Michelle's guidance, Julie was with us to begin with, but with Michelle's guidance, we also understood that we want to make it our own and make sure it has our values attached to it. And so that's when we started using reciprocity, reverence, respect, resilience, and responsibility. So those were the values that we got working with. We had Shirley Trahan help us get those into Salish translations and help us define those words for us based on what she believed that they meant. And so all of that's been, you know, encapsulated into these lessons. We work on Julie's calendar that she developed many years ago for place-based Salish rounds. It's the calendar, the vocabulary, the words that go along with that, and that those are introduced to the children every day. We have lessons that focus as much as interaction as we can get. And I know they gave me my 1-minute sound here, but so we had also Jennifer Finley wrote a book called Huckleberries, Buttercups, and Celebrations, and we gifted all of the 4th through 6th grade that second year with that book. And so, you know, just really trying to work with the different departments with the tribe too. Patricia Buffalo helped us with some of the mental health understandings of what it was like as, you know, a young girl growing up, you know, and I'm her age, but to say what it's like to be able to know when to ask questions and how to think about developing questions based around your emotions and family values. And so all of those things, Willie and Patty Stevens were extremely instrumental when I wrote the curriculum on hunting camps and gatherings. And I don't know if I said Adele Martin, but she was really instrumental in helping us stay positive and make sure it was an asset-based so that we didn't get bogged down in heavy emotions with the kids, but we're just giving them really solid coping skills. Okay, I can quit talking now, but love being here. Thank you.
Okay, I'm gonna try to do rapid fire, I guess, because we're running out of time. I just want to introduce myself again. My name is Sarah Big Sam. I work for the Tribal Education Department, and I have recently been appointed by Michelle Mitchell to be the Tribal Education lead on this project. And I just want to leave you with this thought. I have a deep connection to Dixon and Shalit, but I feel like they're in the middle of nowhere on the edge of everything, right? So where we're going now is really just working on building those relationships. And so I'm really excited that our CAB is growing. We finally have parent members on that CAB. We're getting voices, we're getting ideas, and we're really excited to see the tangible things moving forward. What is it going to look like in this community so that we can build that relationship and help Shalit feel like a strong, solid community. We might have a couple minutes for questions if there is any that you said of them.
Any questions from council? At this time, I would probably open it up to our audience if there was any questions. Because it's a moment to share and learn from everything that we have going on. So, yeah, go ahead, Danielle.
No, I just want to thank you guys. It's just been a pleasure to work with you all, and it's just, yeah, it's amazing, and I'm looking forward to what's to come with this project, and I think it's going to have a really positive impact on the school and on the children in our community. So thank you for your hard work.
I wanted to say a personal thank you. Your compassion, passion, and work ethic is incredible. The community is incredibly lucky to have you. So thank you for your continued support.
Thank you.
All right. So thank you, Dr. Anissa and her team. I just kind of got going on everything. So I guess a little introduction. This is our first biannual research update symposium. We expect all researchers to come in and present annually. And so this is just kind of a way to get the community involved. We're all impacted by these different research projects. So we're trying to be more transparent with the research projects. And so each presentation will be 15 minutes, 10-minute presentation, and 5 minutes for questions. We have 3 presenters on Zoom. So our next one is on Zoom, and it is with Shannon Hilty. You can say if you want. Let me double check that she is available.
Can you hear me?
Yeah, we can hear you.
Awesome. I am here.
Okay, go ahead.
Awesome. Let me share my screen really quickly. All right. Can folks see that? Okay.
Looks great.
Awesome. Well, thank you. Hi, everyone. My name is Shannon Hilty, and I am the bat biologist for Montana Fish, Wildlife, and Parks. And I just want to start out by saying, bats obviously fly, right? They do not care about county or tribal or state or even international borders. And so I personally think they're a really good reminder that, conservation efforts really rely on partnerships and conversations between communities. And Montana Fish, Wildlife, and Parks and myself are so grateful to have the Confederate Salish and Kootenai tribes as partners. And I just want to take a moment to thank the Tribal Council, the Tribal Research and Review Board, especially Matea. It is so awesome that you guys are doing this, and I'm excited to be part of the first one, and also the community for helping us and partnering with us on some of this research. And then I also just want to shout out to Carrie, Kaylee, and Kara. They have been instrumental in getting these projects off the ground and doing a lot of the field work on CSKT lands as well as permitting. And so, yeah, again, just thank you so much for having me. Okay, so I'm going to give you kind of a 30,000-foot view of one of the largest ongoing non-game projects we have right now. And at the end, I'm just going to give you broad things. So if you have questions, feel free to let me know. We know that bats are important. However, they face a lot of conservation threats. White nose syndrome is a disease that's caused by a cold-adapted fungus called Pseudogymnoascens destructans. We'll call it PD for short. And so what happens is essentially this fungus gets on the skin tissue of hibernating bats. So you can see it here on the muzzle of this bat. It's all along the wings and on the membrane of the tail. And what happens is that the fungus starts eating through the skin tissue, which is really painful, and it causes bats to wake up repeatedly through hibernation. And they only have a set amount of fat reserves to make it through the season. And so what happens with the white nose syndrome infection is bats wake up a whole bunch and essentially they end up starving to death by the end of the season. And so I do want to just point out that not all bats are affected by white nose syndrome. Some bats just carry the fungus and never see symptoms of the disease. And then the bats that are impacted by the disease, they suffer varying degrees, I guess, of symptoms and severity. So we've been watching white nose syndrome spread in North America since 2006. It was first detected here in New York State. We've been watching it spread westward. And since its discovery in North America, it's driven sustained and really significant population declines in some species. Some species are seeing 90 to 98% declines, so really serious declines. And so our question, as we've been watching this progress, has been, how is it going to affect Montana's bat populations? And so to get at this, partners in Montana began collaborating on a project to systematically assess both the timing and the distribution of the fungus and the disease in Montana. And we coupled that with the North American Bat Monitoring Program to assess population trends. And I don't have a selection, but for this disease component, and at those sites, so we kind of have a variety of sites. So we have sites where we know bats are hibernating. So we'll go into those hibernaculum and we will swab bats like I'm doing here in this picture. You can't really see it, but there's a little bat here on this cave wall. We also, in the absence of a lot of known hibernacula, we will go and catch bats out on the landscape using different tools, like the mist net shown in this photo here. It's hard to see, which is the point. But then we capture the bats. When we have them in hand, we swab their noses and their forearms, and then we also look for symptoms of the disease. So this is just one example of that. When bats have really bad infections, those lesions glow this really bright yellow under a UV light. So this bat has a pretty bad infection. And just to give you kind of a broad overview of what we found in the state. So we ramped up our sampling efforts in 2019, 2020. And we had our first detection of the fungus, which is these little red dots in 2020, followed by the disease, which are these black dots in 2021. And since then we have watched it spread westward across the state. We do not have the 2025 results summarized yet. We're working on that right now, but I do just want to point out that we did just have our first detection of the fungus in Lincoln County, Montana. This is the first detection of the fungus west of the Continental Divide in Montana, so that is pretty significant and quite a bummer. Lick Cave, which is this little green dot here on CSKT lands, still remains PD and white nose syndrome negative, but I expect with this new detection that we're going to see that change in the next year or so. Okay, so we're tracking the distribution and the timing of the disease and the fungus across Montana, but we also partnered with the North American Bat Monitoring Program, or NABAT for short, to monitor population trends in relation to threats like white nose syndrome. And so one of the nice things about NABAT is it provides this spatially balanced sampling framework. So this is what it looks like in Montana. And from that, we can randomly sample these spatially balanced samples across the state. And that's what these bolder dark blue cells are. And another thing that NABAT does is it provides standardized survey types so that we can all do the same thing. So data are comparable across projects. So Montana participates in acoustics. We do external colony counts and internal colony counts. I'm only going to talk about acoustics for sake of time. This is a huge effort on everybody's part. So crash course in bat acoustics. So bats can actually see pretty well. Bats being blind is actually a myth, but they can't see very well in the dark. And so bats send out these high frequency echolocation pulses to navigate and find prey. And we can put these high frequency microphones, these specialized microphones out in the field, couple it with a bat detector. Bats fly around the microphone all night long, and we record those echolocation calls and then bring them back into the office, and we can learn all things about species composition, bat activity, foraging, et cetera. So it's a very cool passive tool because we don't have to touch the wildlife, which is like the pie in the sky dream. That's what we want. So just kind of a little bit more detail on the methods. So for these acoustic surveys, within one of those 10 by 10 kilometer cells, we put out 4 detectors and we let them sample for 4 nights. And that helps us assess species present within the cell as well as things like activity, foraging, et cetera. And just kind of a broad overview on some of the bigger picture things we're seeing. So we now have 6 seasons of data. We sample anywhere between 80 to 122 cells per year. That depends, on partners and how much they can take on and how much funding I can get to hire bat techs to kind of cover the state. And we've recorded over 4 million call sequences to date. So it's a huge data set. And I want to give you just an idea of what it looks like on CSKT lands. Because I think it's really cool. So Lick Cave is up in here. This is the hibernaculum that we survey annually. And then we have 2 NA-BAT cells that are surveyed by CSKT biologists annually down here. And so those have been sampled between 2021 and 2025. And we've recorded over 34,000 echolocation sequences. So it's a very robust data set just on CSKT lands. And we've had experts go in and hand review recordings to identify species definitively. And we've identified 9 species across those 2 sites, 5 of which are susceptible to white nose syndrome. And so this is a very important data source for us. You guys have a huge data set. I think there's a lot of cool stuff that we could do with it. I do want to talk about just really briefly some of the bigger picture things that we do with these data. There's a lot going on here. I think this has been one of the most fun parts of my job is to actually see where the rubber meets the road. But I don't have a ton of time to get into details. So I'm going to talk about these 2 things. So we did partner with USGS to model basically the impacts of the fungus and the disease on our bats. And they did determine that there was a detectable but negative effect of PD on our white nose syndrome susceptible species. So that is not shocking. But now that we have, I think, 3 more years of data, we're going to continue to rerun those models. And I think where it's going to get interesting is when we get to the western portion of the state where bats aren't really hibernating in caves. They're hibernating in maybe talus slopes, rock outcrops, cliffs. We don't know how the fungus, disease, or bats behave in those features. So this is a really cool project. And then another thing we're doing with these data is, looking at these internal colony counts and declines. So like when we go into Lick Cave, we count all the bats that we see, right? And we have noted now 90 to 98% declines in 5 of our largest known hibernacula in Montana that are white nose syndrome positive. So that is not great. It's really a bummer, but it's also not unexpected. So again, I think that silver lining comes when we think about, well, what's going on in these other features. So more to come on this. And then one other thing I want to highlight, I know I'm out of time here, but I just want to point out that we have been working with partners to determine how we want to respond as a state to white nose syndrome. And CSKT has been involved in this process. And one of the cool things that came out of this decision-making process was that we identified management actions that we think are most likely to benefit bats in Montana. So we identified vaccinations and probiotics as likely being really beneficial. I think there'll be some really exciting things coming out on the vaccination front in Montana soon. But we also identified habitat actions such as installing artificial drinking sources, maintaining healthy forests. And we've talked about types of protections that we maybe could offer bats, like protecting maternity roosts. There's no like one-stop shop for this. We're going to have to use kind of a broad suite of actions, I think, to help our bat populations recover. But that's been a really cool collaborative process. And with that, I'm going to hand it over to questions. But again, I just want to thank everybody at CSKT for having us. This really is what successful partnership looks like. There is no way that we could do it without everybody's help. So we really appreciate your partnership. And I appreciate having the opportunity to talk to you all today. And I think we might have time for questions, but I'll hand it over to Matea.
Thank you, Shannon. Does anyone have any questions?
Got one question. Shannon, as far as places where they're roosting, we put up numerous bat house structures. Is there any study specific to the benefits of artificial sites versus the caves or big fir trees or whatever it might be?
Yeah, that's a really good question. So it's kind of a complex answer because we have 15 bat species in Montana, and 3 of our species really, really like artificial roosting structures. So little brown myotis, Yuma myotis, and big brown bat. And all of those species are heavily impacted by white nose syndrome. So they're the ones you're going to find in bat artificial structures. So on that front, I think they're a really positive thing. And the other thing that we're finding, like I didn't get to get into it, but we are really struggling to find bats in the winter. That's been a really hard thing to do. And so we've kind of shifted our focus to bats in the summer. So when we're thinking about like probiotic treatments or vaccines, those really have to be administered at these summer roosts like you're describing. So those are great. And then we have other species like the long-eared myotis. They primarily roost in trees, so they wouldn't use an artificial structure like that. So it's tricky, but yeah, that's awesome.
Mr. Chair,
Yeah, Carol.
did you say what the cause of the disease is? What caused?
Yeah. Oh, sorry. Yeah, I kind of zoomed through it, tried to pack a lot in there, but it's caused by a fungus, and it's a cold adapted fungus, and it just so happens to survive in areas that have the exact same temperature and humidity requirements that bats like. Um, so, we're not entirely sure how it got to North America. There's been a lot of conversation about it coming over in cave gear. But it's since spread across the country. And so one of the things we don't understand is how that fungus that causes the disease behaves in, say, like a rock outcrop or a talus slope. And since Montana's bats are not really hibernating in caves in the numbers that we would expect them to be if they were all using caves, that I think is a silver lining. That's the hope I'm holding on to, that maybe things will be different for those populations. But it's a fungus.
Climate change or something like that, but yeah. Okay, thank you.
Yep.
One more question. If there is a colony that has the white-nosed bat syndrome in it, has there been is there any strategy just to eliminate that colony to prevent further spread?
So when we embarked on that decision-making process that CSKT was a part of, that came up in conversation because we do it, I think, I shouldn't say we, but I know it's been done in other species. But the problem is bats fly and they also spread it, in the cusp seasons. So by eliminating like one impacted colony, you really can't, it doesn't do anything. You're just gonna have more bats show up there the next year. And it survives in the environment really well, which is just another reason why it's been so tricky to manage. So no, unfortunately, that does not work. And the thing with vaccines, I think the promising thing there is like for some species, like the little brown midas, so the species that would use a bat box or an artificial structure, once that population is hit by white nose syndrome, we start seeing them tank really quickly. And so the idea with the vaccines and the probiotics is that maybe you buy them 5 years, you buy that population 5 years, and they develop natural resistance and they don't completely, die off in an area. And then, hopefully they can survive long term, but it's been really tricky just because of things like that.
One last question. Have you been successful, or how would you rate your success in cavers, those people that are recreationists that like to explore, say like the ice caves, or what do we call it, Lick Cave up here, or old mine shafts like in Rive? What is it successful, or are they responsible cavers, or?
Yeah, that's a good question. So that's been really interesting in Montana too. So a lot of states, as soon as white nose syndrome, became a thing in North America, states just started locking caves down preemptively. And Montana went with a different approach where we have been working really closely with our cavers because we do have a lot of caves in Montana and a lot of them are pretty technical. So we have worked with them to actually do extensive surveys in the state. They have actually helped us with our bat surveys, which is great because I'm not good at caving. I do not like it. It's very scary. And they're great. And in return, we get to go to their annual Montana meetings and present and talk about things like decontamination. So I've had really, really good experiences with our local caving communities. But there's always, some people that aren't going to follow, the recommendations. You always get that. But I also think, there's so much movement with our bats that I think bats are naturally spreading it around as well. So it's hard to say.
All right. Thank you, Mr. Chair. Thank you, Shannon.
Wait, Vernon? Any known effect on humans, Shannon?
Nope. That is one silver lining. White nose syndrome only impacts bats, so we haven't found it on any other type of species, definitely not on humans. And the other thing, as soon as bats start coming out of hibernation and they're more active on the landscape, the fungus does not survive really well in the summer, doesn't survive into July, August. So they come out, the ones that survive, and they'll go into like those bat boxes and they'll start recovering. So even those bats that have survived an infection, they may or may not have a chance, but yeah, it does not affect humans or any other sort of animal.
Even vampires? No, I'm just talking.
Well, that remains to be seen.
No one's on the vampire. Any other questions from anybody?
All right. Thank you again, Shannon.
Yes, thank you so much. Thank you to the Tribal Council. I hope to be there in person next year, so thank you.
Good job.
All right, we move to our next presenter. So it is Dr. Clay Comstock with the Department of Life Sciences at Salish Kootenai College.
All right, thank you, everybody. Glad to be here. I've been to Tribal Council now. I think this is my third or fourth time, so pleasure to see some of you again. So as Matea said, I work for Salish Kootenai College in life sciences. I teach chemistry and environmental health. Oh, sorry, gotta. Okay. Go back here. Okay, so a little technical difficulty there. I'm going to talk to you today a little bit about our air quality study that we've been doing. This is one of a couple of studies that we've been working on over the past few summers. So I'd like to start by acknowledging some people. First, the previous CSKT Tribal Council and Shelley Fyant, who were the first to approve this for study. This actually happened before the Research Review Board. So again, I thank everybody for inviting me for this. I also want to thank the students at SKC. These are three individuals who were participatory in this project. Ms. Shay Brown received her Bachelor of Science in Environmental Health. She's just finished with a Master's in Public Health at U of M. Michelle Toller is a secondary education. She now teaches down in St. Ignatius at the high school there. And a recent graduate, Andrea Hall, who's currently accepted to go to PA school. So all three of these individuals went to school here and were instrumental in this project. Without them, this project would have probably not went as far as it did. I also need to thank my collaborators at Montana Tech, their graduate student, Mary Ortgies, who is also doing a follow-up study that's not talked about today, and looking at indoor air quality. And my collaborators, Julie Hart and Dan Attenruth. Okay, so what I want to really do is kind of present some of the data that we've submitted for publication and then sort of open it up for any questions that might be around. So we all live in the valley, or most of us live in this valley, and we know that there are smoky days during the summer. This is a picture in Polson looking at the Mission Mountains, and you can see the mountains here behind a little bit of smoke. But on a different day, it can look like this, where you're socked in, you can't see anything, and you know that when you breathe it in, that's what you smell is the smoke. And so that smoke contains a lot of different particles, depending on what material is actually being burned. The particle that we're most concerned with in air quality right now is what we call particulate matter 2.5. So previously, people were interested in particulate matter 10, which is a larger particle. And these smaller particles actually have been shown to get into your blood, get into even to your brain, and have poor health outcomes, which can also include mortality. So it's the current standard for understanding air quality and human health. Can't really see that. That's okay. So just a few studies that I want to talk about a little bit about why we're interested in this. One of the big ones is that the current estimate is that wildfires in the US alone will cost us $400 to $900 billion a year. And that number will probably go up as more people encroach on what we call the wildlife urban interface. So in particular in Montana, there are a few studies that have been done that talk about wildfire and human health. One of them involves tribal communities. So a particular paper came out a few years ago that showed what we call a community vulnerability index. And all of this is a fancy term that talks about socioeconomic status and healthcare and all of these different things. And ultimately what part of this study showed that where tribal reservations tend to fall within these indexes. And so they're more susceptible to certain kinds of things like wildfire as the basis. And another study showed a couple of years ago that if you average that PM 2.5, those small particles that can get into your lungs and into your blood, that can increase the association with things like influenza later in the year. So fires happen mostly in the summer, but they result in healthcare issues down the road, is the take home there. And so we thought one of the ways that we could contribute to this is to look at these low cost air quality, what we call sensors. And there's a lot of studies that about there. Many of you might even have them in your homes now. They've generally been proven to be pretty satisfactory at measuring these small particles, importantly over long periods of time, and especially in communities that might be disadvantaged where having expensive machinery and equipment and infrastructure to support that can be quite challenging for communities. So what we decided to do was try to build a community-based air sensor network. And so we piled about 6 to 8 of these and evaluated PM 2.5 levels over 3 wildfire seasons, 21, 22, and 23. We didn't look at non-wildfire as well, but that is also something one could look at. So I want to take a minute here to show you this map. This is a map that shows you the rough outline of the Flathead Reservation in red. And as you can see, there are orange dots that are on the perimeter of that. These are the current air quality monitors that the state and the government use to monitor air quality. And you can see some of them are quite far, more than 2 or 3 hours from tribal boundaries. In the center here, there's one marked E. That is a site that's where all the signaling towers and stuff are outside of Pulse and up on Jetty Hill. It's called the Improved site. And it measures air quality, not by us, but by others. And that site measures all kinds of things, including one that we'll see in a minute that's total carbon. So anytime wood burns, it burns carbon and that ends up in the atmosphere and it can be detected. That site is a nice site, but the problem is nobody lives up there. So it's not very useful for sort of normal air breathing. So we decided to put in these community-based sensors, and that's what you see in blue and purple. So the ones in blue are from a different commercial company, and the ones in purple were added later to see if we could build a network, add to that network, and analyze the data in bulk. And so you can see all the sites here. Like I said, there are about 7 or 8 of them in operation during the wildfire season. And these are at people's homes under sort of normal conditions in which they think would be the best place to put these. We don't give them many guidelines. We just say, here it is, put it at your house where you think it would be the most effective. And that gives us that sort of variability in what people do and what they expect and all of that. So the important part here is that we looked at that improved site because it has longstanding data, over 20 years worth of data, and looked to see what the carbon in the atmosphere was around the Flathead Reservation. And that's shown in this graph. And so you can see basically over a 20-year average for between January through December, your largest variation and your most carbon that gets put into the atmosphere is July, August, and September. And that constitutes the standard fire season that most of us know, and in terms of burning and practice all of that on the reservation. And so that just gives us justification to say these are the months that we really want to study air quality. So the take-home message for this data so far is that there is a very good agreement between our air sensor network, those are the air quality monitors that we use that are commercially available, and the regional air monitors that we find outside of the reservation, which are run by EPA and federal agencies. So this is just 1 year of data, 2022, looking at July, August, and September. And what you see here are the daily averages for all of those sensors in purple, and all of those are those 4 air monitors in orange. And again, the idea here is just to show you what some of this data looks like, so you have an idea of what it can do. There are just a few take-homes here. We've done this again for 2021, 2022, and 2023 for July, August, and September. So we have a total of 9 different months across 3 different years. And the data analysis tells us that the correlations between the 2 systems are actually pretty good, considering where they're located and how they're set up and all of that and maintained. We do see a little changes here. So if you notice that the orange lines tend to be a little higher than the purple lines in general. That tends to be because most of the time that we were studying wildfire, there wasn't many fires on the reservation. So that smoke was actually coming from outside in. And so we saw higher peaks on the monitors that the EPA would use. On occasion, we might see a fire on the reservation that might change that. We also sort of see in some cases where the orange line is sort of to the left of the purple line, and that is a sort of a lag in which we're detecting smoke coming in from the outside, is sort of the interpretation there. So another way to look at this data is to say, okay, well, let's use what's called the air quality index. The air quality index is what you would find on the news. When you listen to the news, they say the air quality is good, it's a purple day, it's a red day, it's a green day, kinds of things. And so what we decided to do was use the prediction models that the EPA has to measure air quality index. And we did that centered at Pablo here in town. And so that index, again, by the EPA is shown here. So a good day is green. That's usually less than 9 on the scale, and this is measuring particulate matter in particular, and then 35 for moderate, 55 for unhealthy, and very unhealthy and hazardous. And so many of you have probably seen this index if you watch the news. So what we did was say, okay, well, we don't have air monitors on the reservation. What do the EPA say the air quality would be in this region? And so we then took our air sensor data and mapped it relative to what would be here. And so you can see that, for example, in the green region, those would be the expected values. Those are the values that the air quality monitoring system from the EPA would say based on their data. The purples are our sensor data that we put out. And what we're looking for here is whether we're looking at a good day, a moderate day, a sensitive day, or unhealthy day, we would basically see no difference statistically. And so that says that we are able to capture essentially the same kinds of data that the EPA monitoring systems are capturing, which is ultimately, I think, a good thing. We saw the same trends for both 2021 and 2023 as well. Again, just showing you the 22 data. So just some quick conclusions and limitations of this particular study. Again, this was a community-based approach. We were able to capture PM 2.5 levels fairly accurately across 3 different years. Some problems that we ran into early were recruiting community members during that initial fire season. We thought perhaps this might have been related to COVID or coming out of COVID at that particular time. The western part of the reservation has less people, so picking up people in that side of the reservation is a little harder to find. Internet services there are a little different. Some challenges with using one of these sensoring systems is that if a person puts it next to their grill and decides to barbecue for 8 hours, we're going to pick it up. If they live next to a dirt road, that particulate matter will show up, and also fireworks. So 4th of July, for example, when people are lighting fireworks off, I can tell, at least in the major cities on the reservation, that it's 4th of July. We were able to, in essence, recapitulate the off-reservation air monitoring system. Under those low-level conditions, less than 10 parts, we saw a fair amount of variation. That's not too concerning because that's the good region according to the AQ index. And so nobody really cares as much about that level. In this particular study, we never actually saw unhealthy or hazardous levels on the reservation, so we don't know how these sensoring systems will work at the high levels, especially over long periods of time. And again, I mentioned this earlier, we saw sort of lower levels or lagging behind the monitoring system, so that likely because it was coming from outside the reservation. This system worked really well for determining what we call ambient PM 2.5. And so that's those large smoky days that roll in and stay socked into the valley. They're more dispersed, and so all the monitors pick them up very similarly. It was much harder to capture PM 2.5 that was produced and then didn't hang in the valley. So a couple of instances like some of the fires that were just north of Polson, smoke literally went straight up over the top of a sensor and went over the mountains, and the sensor thought it was a good day because there wasn't enough being produced. It wasn't staying in the valley enough to see it. So there are some limitations there. One nice thing is we did both. What the data you saw was actually corrected data with a system that covers the United States for large smoke. But we were also able to recapitulate this using just the raw data straight from the sensors without any modifications. And so that was nice to see as well. The last bit here is that these sensors actually will capture data every 10 to 15 minutes rather than every hour. The problem there is that the data becomes very intensive to measure things over 10 minutes and for us with students have them be able to analyze that data effectively. So we stuck to a more sort of like hourly, daily sort of approach. With that, I thank you. And if you have any questions, I'd be happy to discuss them.
Question in the back. Go ahead, Mike.
Hey, we need to meet. Last couple of years, played worked by 40. Per player one. And we have them in almost every school or outdoor. I think almost every fitness center, and the health center. We're getting more, so get player.com. Full-time air with top awareness for folks. We do, all that stuff. So let's get together and talk about.
We're planning to come in and do some trainings. We need to increase the communication between facilities managers and occupants of buildings and the people who are monitoring the sensors, the human aspect of this. Because we did notice elevated indoor air, decreased air quality in many tribal buildings, including this building. So we need to work on that. It means making sure your HVACs are functioning properly. It means shutting down outside air intakes, which all newer buildings have. And those things have to be acted on, or you're breathing the same air that's outside. So we'll be in touch in the near future. Any other questions?
Thank you.
Thank you.
Thank you, Clay. All right, our next presenter is Melissa Maggio with the Montana Biocontrol Project. She's the coordinator for that out of Missoula County.
Hi, everyone. Thank you so much for having me today. As I was introduced, my name is Melissa Maggio, and I'm the coordinator for the Montana Biocontrol Project, which is housed at Missoula County Department of Ecology and Extension. I will be presenting to you today on flowering rush biocontrol. So giving you a little bit of an update on what's happening with the development side of it and what's happening on the ground here in Montana, and specifically at one location on CSKT lands. So in case you're not familiar with flowering rush, it is a non-native, aggressive, freshwater invasive. It's capable of rapidly colonizing wetlands, lakes, slow-moving rivers, canals, and irrigation ditches. It can grow both emergent, so it has its like roots in the water, but the above ground part comes up out of the water, or fully submerged, so all of it's underwater. There's both a triploid and a diploid. So those are different genotypes. So they're all flowering rush. The only reason that this is important is because the triploid does not produce viable seed. So it can't spread by seed. And that's what we have here in Montana. There is a diploid that produces viable seed, but that's mostly found on the east side of the country. So our flowering rush here in Montana disperses pretty much, exclusively through rhizome fragments. So you can see on the right-hand side there, the second picture up from the bottom, that kind of shows you what it looks like below ground. So fragments of that, and also root bud fragments is how it's spread. And it comes up really easy. So like propeller motors can easily pull it up and then drop it in new locations on the lake or rivers. So current management options include herbicide, covering, hand pulling, digging, diver-assisted suction, and dredging. However, management is difficult even with all of these options. There are some herbicide programs going on here on Flathead Lake. However, herbicide cannot be used in all locations. It's pretty specific where it can be used. So we need different tools for different locations. Currently, the uppermost population of flowering rush in the Columbia River Basin is found here in Montana, in and around Flathead Lake. It was discovered in Flathead Lake in 1964. There was a big mapping effort that I believe was headed up by SKC in 2008. And at that point, they identified 2,000 acres infested with flowering rush. Pretty significant infestation. It's also dispersed up the main tributary and all the backwaters of Flathead River, as well as the Clark Fork River. And the Clark Fork River feeds Lake Pend Oreille in Idaho, and it was first found in Lake Pend Oreille in 2007. I think it was first recorded. I think it had been there longer than that. But because We are the uppermost population in the Columbia River Basin. It's really important for us to be a good upstream neighbor to our downstream partners, because in a sense, we are where the majority of the flowering rush is coming from for our downstream neighbors. So we're trying to do everything we can here in Montana to stop that spread downstream. So because of the difficulty in control, we formed a biocontrol consortium in 2013. A biocontrol consortium is basically just a group of like-minded stakeholders who have a common interest in developing a biocontrol system. So in this case, for flowering rush. And they pool their funding, and all that funding works together to go overseas to our research partners in Switzerland for them to help us identify potential biocontrol agents and test them. We were pretty excited about flowering rush as a candidate system for biocontrol because it is the only plant in the Butamaceae family. So often when we encounter troubles in the testing process with our potential biocontrol agents impacting something native or beneficial, it's closely related to our target species. So flowering rush does not have any close relatives. So that was definitely a plus in developing a biocontrol system. So the first thing we did was identify our genotype, which I mentioned to you. So you can see on the bottom map there on the right, those are all the different genotypes that we have here in North America. And so the reason that we did this is because often these biocontrol agents that we develop are so host specific that they will only attack one genotype. So we wanted to make sure that they attack the genotype that's invading here in North America. And then the next thing that happened is our overseas research partners, CABI, they go out and they do native range surveys. So they look to see what is attacking flowering rush where it's a native plant. And then they bring that back into the lab, they do literature surveys, and then they do host specificity testing, making sure it only negatively impacts our target weed and also that it's impactful. We don't want to release something that's not going to actually help us achieve our goals. So the three little inset pictures you see here are kind of the stars, the ones that rose to the top that we worked on developing first. So Bagus nodulosus is a weevil, Phytoluriomyza is a fly, and Dysontia is a white smut. So I'll tell you a little bit about each of those. Oh, sorry, it looks like my font got messed up. So a little bit about the fly. We're pretty excited about this one because it's showing some pretty significant impacts to the plant. So they saw a 50% above ground biomass reduction when a plant was exposed to just one female within 2 to 3 weeks. And my second to last bullet needs to be updated. They've actually conducted host specificity tests on all 40 of the test plants, and they have not seen any impacts to anything but flowering rush. So they're currently preparing the petition. So when we do this research, our overseas partners do this research, we don't bring anything into the country. We want to make sure it's safe before that happens. So they do all this work in Europe. They summarize all of that research and they put it in a package that we call a petition. That's submitted to USDA APHIS, who's the regulator of biocontrol in the United States. And it goes through this regulatory process. U.S. Fish and Wildlife Service is a part of it for Section 7 review. There's also a tribal review. And then if it gets through all of it and it's approved for release, then we get a permit to release it in the field. So the next is this smut. So this is much earlier in the development process, but they have identified a strain of this fungus that does attack our genotype, and it attacks fully submerged plants, which is incredibly important to us because you can see here in this video, I took this at Finnan Slough, that this is a significant infestation of flowering rush that's growing fully submerged. And most of our insects will not be able to attack infestations, this large of an infestation fully submerged because they do breathe air. So this will be a really important tool in the future. And then the last one I want to tell you about is the weevil. So this one, I'm running out of time, so I'm going to kind of skip a lot of these details, but I'm happy to answer questions. This has already made it through all of the testing, and it showed no impacts on anything but flowering rush. It was approved for release in the field in Canada in 2023, and they actually made the first releases of it in 2024. It was approved for field release in 2024 in the United States, but we have not released it in the field as of yet. I'll tell you more about that in a moment. So on the ground here in Montana, the first thing we did was beginning in 2022, we were trying to identify ideal sites to utilize this biocontrol agent. And so we were looking for kind of some specifics for ease of access for us, where we thought it was more ideal for this weevil, where we could monitor. And so we identified 2 locations. One of them in 2023, Ducharme Fishing Access, and the other we identified in 2024, which is at Finnan Slough. So generally, this is where the 2 locations are that we've been pre-release monitoring for the last couple years. So we developed a monitoring protocol, which is something totally new to us. Most of the people I work with in biocontrol, we work pretty exclusively in terrestrial systems. So this was pretty new to us. Monitoring in an aquatic system is much different. So we developed this protocol. This is basically the data that we're collecting. We're collecting percent cover from the water surface up and water surface down. We'll also eventually be collecting below ground biomass samples as well, because that's something that we really need to decrease and what we expect to decrease from the biocontrol agent. That's where all the plant's root reserves are. We also this summer, you can see how happy I look there to be in my wetsuit in Flowering Rush. We also this summer collected some soil substrate samples to determine what macroinvertebrates are utilizing the substrate in Flowering Rush infestations and also outside. Those are still being analyzed by an entomologist at ARS. So some additional things that happened where I got to go to the quarantine lab in Sydney to learn how to sex the weevils, put them in oviposition cups, because they have to be reared through 1 generation in a quarantine before they can be brought outside of quarantine. So the insects that come to the United States from Switzerland must be reared through 1 generation. Here's a weevil swimming too. It's pretty adorable. Isn't that just so cute? And then we have these rearing ponds at the facility in Missoula. And so our intention is to release weevils from the quarantine into these ponds to really build up their numbers before we start utilizing them to manage the wild infestations of flowering rush. The last thing we did this summer is when we first put these weevils out into these infestations, we want to cage them for a short period of time just to ensure they can find one another to mate and lay eggs. This is a huge infestation of flowering rush. We're worried if we put 25 weevils in this infestation, they'll all go different directions and they won't mate and lay eggs. So then we won't have a population that's growing. So plans for the future is we're going to continue this annual pre-release monitoring. That's really the only way that we're able to determine if we're seeing an impact from this biocontrol agent. We will be taking soil cores. Potentially we'll continue to take these substrate samples to look at macroinvertebrates, maybe going to Switzerland to help collect weevils, potentially working in the lab again. There's been a lot of issues with our federal partners with funding and hiring freezes. So we want to provide as much assistance to them as we potentially can. It's a long list of partners that help to make this project happen. And I would like to thank you all for your partnership in this as well as everybody else. There's also a lot of funders. This is another way we partner, and these projects would not happen without the on-the-ground partners and the funding partners. So thanks to everybody. All right, and if there are any questions.
Lisa, I had one question. As far as native rushes, what is you're saying that you haven't identified any secondary effects or negative effects on any native plants?
Right. Yes. So we have, like bulrushes and carex, and all of those things that are native, and those are actually in a different family than flowering rushes. So they're actually very distantly related, even though they look pretty similar and grow in similar areas. But no, they've tested all these biocontrol agents against a variety of those beneficials as well. And they've seen absolutely nothing negative happening on any of them.
Have you done anything as far as fish species that might feed on these aquatic?
So this is a really tiny weevil. It's probably like a quarter of your pinky fingernail. And it's host specific to flowering rush. So without flowering rush, it cannot survive. So no, we have not specifically looked to see if any fish are going to eat this weevil, but I can tell you that this weevil's population will never build up to the point where it's going to be a significant food source. And if it does its job and it eliminates flowering rush, there'll be very few weevils in the system anyways.
Some of these areas that have flowering rush also have non-native fish species.
Yeah.
Warm water fish species.
You've heard that.
I don't want to feed them with something else, but
Yeah. I mean, that would really be a bummer for us too, if that does happen. And it is absolutely a concern. We're also concerned about like dragonflies and things like that feeding on them.
Question in the back. Go ahead.
Thank you. This is just is this working? Thank you. Okay, just a quick general question. So, as the project continues, and if there's full success and this plant is now like dead in the water, I'm just curious, is that something that has to be pulled out? Is that something that just goes back to the environment? For projects like this, I don't know, I guess, what success looks like and something to prepare for the future.
Yeah, that's a great question. So when I'm talking about biocontrol, I'm talking specifically about classical biological control. And so what that really does, success from classical biocontrol brings a balance back to the system. So it's never a complete eradication of anything from the system, but it does significantly reduce and stop the spread often of that target species, but it allows the habitat to look more like what it should look like, or natives to come back in and compete with that invasive. It also takes a very long time. So we're not talking about like putting these weevils out and the next year all the flowering rush is gone if they're successful. So a lot of people are familiar with biocontrol for spotted knapweed. And that's a very slow process. Like we're probably talking 8 years before you start to see a decrease in spotted knapweed from the establishment of the biocontrol agents. And that's also a weevil. So we don't really know what to expect timeline wise until we get things out on site in the natural environment. But even with a really fast moving biocontrol system, like for leafy spurge, we're still talking like 3 to 5 years before you start to see decreases. And it's typically kind of a gradual decrease. So there's really probably no reason to have to think about manual removal, especially something like flowering rush. It senesces every year. And so those dead leaves are floating around and doing whatever. So it's not natural to the system since it's non-native, but it's what the system has been used to for as long as that infestation has been there. And I think, sorry, Virgil, just real quick, one more point. Another thing I'd like to point out is my understanding is that at Ducharme Fishing Access, that used to be pretty open, that there was nothing growing there before flowering rush moved in. So it's probably not a concern of putting something back in to replace it. I think that was really meant to be more of an open aquatic system.
Well, there's quite a bit of chara and potamogeton in there.
All the way out as far as, oh, okay.
Comment on
the existing vegetation that was there prior. If you look at the slide there's a fair amount of flowering rush above the water. I mean, it's got a lot of foliage. Underground, 6 times that is what's tied up in the rhizome. So there's a lot of biomass under the ground, under the surface of the water. What do you term that? The bottom of the water column, top of the soil column, something like that. That wasn't there before. So there's a lot of extra biomass growing there. And in 1985, during the goose study, Tom, you remember those days, right? We won't talk about them. Flowering rush was a rare plant in East Bay. And it's very prevalent there now in a lot of the bay. That bay, Elmo Bay, Dayton Bay, and then a lot of sites around the lake as well. So it also, during the goose study, it was acknowledged at that time that they wanted more vegetation in the bay to encourage fisheries, which I think that management direction has been changed now, because that would have just benefited the perch and what's in there now, the pike and the bass. End the comment.
Thank you.
Is your office at the fairgrounds?
It is, yes.
New building there?
Yeah. Yeah.
I'm jealous.
Yeah, it's pretty nice. I feel pretty lucky. And that's actually where the rearing ponds are too. So if anybody would ever like to see them, especially once we get weevils, I would be happy to give you a tour.
Is that on the south side of the building?
It's, yeah, in the gardens.
Yeah, in the garden?
Yeah, like right along the fence. They put me back in the back corner because it's not as pretty as the rest of the gardens, but I still appreciate any space I get.
See any more questions? Thank you.
Thank you so much.
Thank you. All right. So our next presenter is Dr. Jay Carlisle. He is located at Boise State University. He will be presenting over Zoom. And also, if anyone wants any snacks or drinks, feel free to grab some. We have some at this back table.
Are you guys able to see my screen?
We sure can.
Fantastic. Well, thanks, Matea and the full Travel Council for pulling this together, for inviting me to be a part of it. I'm sorry I can't be there in person. And before I begin discussing our shorebird survey effort across Montana for the last few years, I want to just quickly acknowledge my co-authors, Heather and Jeremy, who have done the lion's share of the work to conduct this survey work over the last few years, especially Heather, but also in terms of pulling together the data for this presentation. And I'll also mention all the pictures in the presentation are taken by volunteers and other participants in the survey effort that we've done over the last few years. And let's see, next slide. How do I advance that? Okay. I'm going to start by giving some context for this shorebird survey work across the Intermountain West, and then I'll drill down and look at survey data from Montana, our effort from Montana, and in particular on CSKT lands. I don't know how many of you are bird biologists or bird appreciators, but there's been a lot of recent data over the last few decades that have shown that populations of what we know of as shorebirds, so this includes the long-billed curlew shown in the picture here, killdeer, plovers, sandpipers, avocets, and a whole suite of other wading birds that tend to have long legs and mostly use wetland or lake edge habitats, mudflats, but they also use grasslands to a large degree. Many of these species are in steep decline and have been for many decades. And so there's a lot of concern by management agencies, tribal agencies, state, federal, et cetera, about the status of shorebirds and their habitat. And so the objectives for this Intermountain West Shorebird Survey were to conduct an inventory of shorebirds during spring and fall migrations throughout the interior portion of the Pacific Flyway. And I'll show a map of that over a 5-year period. And then also part of this is to repeat a survey effort that was conducted in 1989 to 1995 across much of the region. And then enable us to then gather current information on distribution and abundance of a number of shorebird species through Montana and other states. And so as this project was beginning, Montana Fish, Wildlife, and Parks reached out to us to see if we were interested in helping coordinate this effort as we've worked with Fish, Wildlife, and Parks on several other bird monitoring projects. And we were happy to get that done. And so we've coordinated an effort over the last 3 years to do surveys across Montana. A little more background for the project. I mentioned there was a historic survey effort that had happened across much of the interior West, but that prior survey effort did not include Montana. So it included Idaho, Washington, Oregon, California, Nevada, Utah, down into Arizona. And the idea behind this current effort is to repeat surveys at all of those sites, but also expand to the rest of what we consider the interior portion of the Pacific Flyway. And so this next map shows both the historic sites that are in blue, the royal blue color, and then new sites to this survey effort in the 2020s in orange. And then hopefully you can make out the dashed lines at sort of the perimeters of this survey area, but basically the interior of Northeast California, the interior half of Oregon and Washington, and then over to Western Montana, and then down is the primary survey area. We agreed with Montana Fish, Wildlife, and Parks that, hey, if we're going to do surveys in Western Montana, let's get surveys done at important wetland sites in Eastern Montana as well. And so we also established some additional sites that extend out to the northeast of Montana. So over the last 3 years, we've done this work in Montana, and then for 4 years, other partners have been working in a few other states. And so you can see there have been over 500 participants across this full region since 2022, a number of sites surveyed as a repeat of that effort in the 1980s and 1990s. And then I'll just mention that Audubon and Point Blue, the two entities shown on the bottom of this slide, are the two leaders of the overall effort. And in addition to coordinating range-wide the survey effort, they're also working to analyze wetland distribution and shorebird numbers from that historic period and now to be able to get these time period comparisons. Okay, and then just one other result that I'll mention is that this is a map that Max and Blake, who are the two main coordinators of the study, shared with me. And these are all sites across the region that have hosted at least 1,000 shorebirds. So these would be like regionally important sites. So at least 1,000 shorebirds during at least one of the surveys, whether it was spring 2023 or fall 2023, et cetera. And 3 of those are in Montana, and that's Bowdoin National Wildlife Refuge, Freeze Out Lake, and then the Goose Lake Complex. And so here's a map of the 19 sites that we, along with Fish, Wildlife, and Parks and some other shorebird experts in the state, selected as the sampling frame to help get some data on shorebird migration in Montana. And this includes National Wildlife Refuge, wildlife management areas, and other wetland complexes and lakes and reservoirs, et cetera. And we've covered much of the state. There's definitely a bit of a gap in that east central and southeastern portion of the state, but we have sites basically spanning east to west and north to south. Our survey methods, this is where we're getting into the sort of nerdiness of the bird aspect of it, but we did surveys, standardized surveys in each year from the end of April and spilling a little bit into early May, and then in the middle of August to sample what are considered the peak migration periods across our region. I will note for anybody that really knows birds in Montana that April window is too early for Montana. So our spring migration survey period missed the peak movement period, but we still did it that way to align with the rest of the effort across the full region. And then we use standardized protocols to survey birds at all of these sites, matching a protocol used in that historic study in the 1980s and 90s, and also matching a protocol that other partners use across all of the Pacific coast from Alaska down to Chile. So we have this comparability of shorebird numbers using the same type of survey approach across regions. And we did these surveys during daylight hours, basically from 15 minutes after sunrise to 15 minutes before sunset. mostly accessed by car or foot, but some sites could be surveyed by boat. And then everybody entered their data into this California Avian Data Center, since that's where Point Blue works with their data. And then that then feeds into the entire region's effort. So here's where I'm going to discuss the results for a few slides, just to give you a taste of what our efforts have resulted in. And this slide shows, just kind of summarizing, numbers and diversity of species across Montana in 2025. And so during our spring surveys, we counted a little over 3,300 individual birds of 23 different shorebird species. And in our fall surveys, we were just under 8,900 birds of 24 different species, for a total of over 12,000 birds. And if we look by season, we see that in spring, American avocets were the most numerous bird that we counted. And then in the fall, the Baird's sandpiper was our most numerous species this fall. And then if we look at highest abundance by site, in the spring, Freezeout Lake hosted the highest number of birds in our surveys. And in the fall, it was Bowdoin National Wildlife Refuge. So some pretty good numbers of birds and good variety of birds moving through Montana. And this is the slide and the next one that I'm going to apologize for, because that's a lot of letters and numbers. And I know this isn't the most efficient way to present some data, but, well, it's maybe most efficient, maybe not the most effective. I'm just going to ask you not to worry about the details unless you're a bird nerd like me, but just to know we're showing numbers by year for all the species that we've detected. And this is for the spring surveys in that April window. And the main thing I'll point out is that in general, the species that were most abundant in some years were also pretty abundant in other years, but there definitely were a few species that showed some pretty dramatic variations or spikes from year to year. And some of that could have to do with weather in a given season. So for example, in the spring of 2023, it was a pretty snowy April. And so I'm guessing that limited the amount of shorebird habitat and therefore shorebird that had arrived to Montana yet. But anyway, this is the kind of numbers that we generated during spring migration. And then this is the next busy slide, even busier, because in the fall migration, we tend to detect a little bit more variety of species. You can see we're capturing quite a number of birds and quite an abundance of a number of species. The one result I'll point to here is in the middle of the right panel. The Wilson's phalarope is a shorebird species that's pretty charismatic. It's one of the shorebird species that will spin in a circle for feeding. And they're well known to use saline lakes, but other wetlands across the Great Plains in the West. They've actually recently been listed or petitioned for listing under the Endangered Species Act because of concerns about dramatic population declines. And you can see we counted many more in 2023 than we did in 2024 or 25. I want to caution, this is a 3-year data set, and it would be not smart to overinterpret this data, but it could say that, hey, yeah, there's maybe even more reason for concern for that species. Or it could just be we missed the peak in the other 2 years and we really hit the peak during that 2023 season. And then to focus on some of the sites where we surveyed with permission of the CSKT, this includes Flathead Lake, some of the sites around Flathead Lake, 9 Pipe, both the National Wildlife Refuge and the Wildlife Management Area, and then Pablo National Wildlife Refuge. I'm not going to get into all the details here, but you can see that of all these sites, 9 Pipe had the biggest numbers, whereas Flathead had the smallest, in part because water levels in the fall sometimes aren't conducive to shorebird habitat. And so in fall of 2025, we didn't detect any, and in fall of 2024, we didn't even conduct the survey just because there was no open habitat that would have been good for shorebirds. But all of these sites had some good numbers of some important species, and those include avocets, killdeer, greater yellowlegs, Baird's sandpiper, Wilson snipe. And so for us, this is data that contributes to Montana Fish, Wildlife, and Parks and the agencies that they partner with for habitat conservation for shorebirds and other species. A bunch of people to thank, including Fish, Wildlife, and Parks for funding this survey and for trusting us to help coordinate it and get it done. Point Blue and Audubon, including Blake and Max, for coordinating and spearheading the entire effort and really providing a lot of support to folks like us to help implement this. I also want to thank the CSKT, including Carrie Kingery, for helping with the permit process to make sure that we could conduct these surveys in these important areas, and also refuge staff at Fish and Wildlife Service refuges for helping with permits, et cetera. And I just want to point out that we had 32 different shorebird volunteers that helped conduct these surveys. We paid their mileage to help get it done. We also hired some staff to help get some of the surveys done, but most of this was a volunteer effort and a lot of enthusiasm. So we could not have done it without all those folks. Some of you might know Stan Center or Dan Casey, retired biologists with a lot of knowledge about shorebirds, but they were really important to getting this effort going in 2023. Here's a slide with some contact information in case folks might want to reach out. I'm going to come back to this in a second. I'm just going to show a few of the references that I cited that look at shorebird population declines or that historic study that I talked about. But I think I'll leave it here and I'll see if there might be any time for questions. And thank you again for your time.
Questions from council? Yeah, I have one question, I guess, in your observations. Of course, climate change is a consideration, bird flu, whatever, loss of habitat from general reasons. But is there any correlation between wetted perimeter of ponds and wetlands and your numbers?
Sorry, did you say wet perimeter?
Wetted perimeter, like the amount, because when I look around the Kettle region here in the Mission Valley, I see a lot of ponds drying up. And so
Right.
certainly the man-made structures where we're controlling the Flathead Lake's one big reservoir, and these other ones are all man-made ones, all the ones you're talking about typically are. But the natural water bodies seem to be in decline as far as the long-term persistent drought effects. So is there a correlation between those habitats and the number decline?
So I don't have that answer yet. I mentioned in one of the earlier slides that Blake and Max are leading an analysis of both wetland area and bird numbers across time periods. And so I'm hoping that within a year or so, they'll have dug into those analyses more, and that might include the footprint in Montana. But I think you're likely onto something. I think there are a variety of causes, including habitat conversion in some areas, but changing climate, drying climate could definitely have an impact.
Well, what makes me concerned is that if we're monitoring places that are altered, the water levels are augmented by our management activities, and that's where you're doing most of your survey work. And we're not out, in the native ponds, the kettles that are, looking at birds there. That actually the forecast of decline might be much greater than it really is.
Right. If we were including some of those small, like you say, kettles or prairie potholes, et cetera. That's a good point. If we were able to manage levels at certain sites and that's what we're monitoring the birds at, we might not get the true measure of decline. I take your point. I don't have a great comeback for that at this point, but I think that's an important consideration.
My cousin Bill Swaney was here. He'd have all kinds of questions.
Well, if you want to share this and have him email me, I'm okay with that. Well, I don't know if anybody else has any questions, but I appreciate your time and sorry I couldn't be there in person, but thanks for making this happen.
Thank you, Jay. All right, we'll move to our next presenter, Megan Ahern. She's with Salish Kootenai College. And so she sent a message that she has some technical issues. So if we lose her, she'll be right back on. Go ahead, Megan.
Hi, thank you. I am having a couple issues with my audio driver, and it only can pick me up if I'm very close to the camera. So I am going to turn my camera off during my presentation, and then I'll turn it back on for the Q&A. Okay, can you all hear me?
We hear you great.
Perfect. So hello, everyone. Before I get started, I'd like to thank the Confederated Salish and Kootenai Tribes, as well as the Salish Kalispel Culture Committee for inviting me to share a portion of my research. My name is Megan Ahern, and I was a graduate student at Salish Kootenai College. I'm here to discuss my development of a relational environmental assessment of the proposed Sheep Creek mine in the Bitterroot National Forest. This research took place in the Woods Creek West Fork Bitterroot River watershed in the Bitterroot National Forest. The forest contains 1.6 million acres in southwest Montana and eastern Idaho, with the watershed comprising just over 36,000 acres. The Bitterroot supports local economies, provides endless recreation opportunities, and produces cultural resources on which Salish Kalispel peoples rely on for the continuation of traditional practices. Currently managed by the U.S. Forest Service, the watershed is the heart of Aboriginal Salish Kalispel territory and was sustainably managed by Salish Kalispel peoples for over 12,000 years. It's also the site of the proposed rare earth Sheep Creek mine. This is not an AI-generated image. This is Mountain Pass Mine in California, which is currently the only rare earth mine in the United States. Between 1984 and 1998, this mine spilled about 600,000 gallons of hazardous waste onto the desert floor. Though the proposed Sheep Creek mine is not planned to be an open pit mine, U.S. Critical Materials, the company exploring the claims, has repeatedly drawn comparisons between the proposed mine and Mountain Pass Mine when discussing operations with investors. The proposed mine has generated some backlash from local communities. Okay, a lot of backlash. As such, it's imperative that an environmental assessment be conducted to establish a baseline of the watershed. Historically, environmental impact statements and other regulatory processes preceding natural resource extraction have been rooted in Western science, completely disregarding Indigenous knowledge and Indigenous research methodologies. However, we seem to be on the edge of a paradigm shift with burgeoning interest in incorporating traditional ecological knowledge, or TEK, in research and management. In 2022, even the White House produced revised guidelines for the inclusion of TEK in agency reports. The location of the proposed Sheep Creek mine, in combination with its significance to diverse stakeholders, presents a unique opportunity to contribute to the shift by conducting an environmental assessment of the Woods Creek West Fork Bitterroot River watershed, implementing both Western and Indigenous research methodologies. I have had the immense privilege of undertaking this work. In developing this work, I pursued two research objectives. One, characterize an environmental baseline of the Woods Creek West Fork Bitterroot River watershed, and two, outline the cultural significance of the watershed to Salish Kalispel peoples. In accordance with guidance from the Salish Kalispel Culture Committee, I will be centering this talk on the first objective. To establish a baseline of the watershed, I chose to explore four key research areas: water quality, the aquatic macroinvertebrate community, the fish community, and rare carnivore habitat. To understand water quality in the watershed, I analyzed data collected through surface sampling conducted by the Bitterroot River Protection Association in September 2025 at 3 sites downstream of the proposed mine. Here, I visualized the results of chemical analyses BIRPA conducted. Unsurprisingly, the water sampled is pristine, with only 7 of 19 targets occurring at detectable quantities. What I would like to highlight, though, are those that went undetected. Both arsenic and lead were undetectable in Johnson Creek, Sheep Creek, and the West Fork Bitterroot River. As shown in U.S. Critical Materials' own data, ore sampled from within the mining claims contains high levels of both. Mining operations would mobilize these contaminants, releasing them directly into the water table. Further, selenium went undetected at all 3 sampling sites, but is commonly associated with rare earth deposits such as those found in the Sheep Creek site. Selenium presents a serious concern for aquatic and terrestrial ecosystems due to its propensity for bioaccumulation and biomagnification. Selenium also competitively excludes mercury in tissues, which can lead to worse long-term outcomes due to underestimated mercury content during environmental monitoring. Shifting gears now to aquatic macroinvertebrates, in August 2024, I conducted 3 kicknet transects in Sheep Creek, each with 3 sampling sites, with the help of my field technician, Mackenzie Lehman. She's there in the crowd today, but is far too humble to stand up. So if you recognize her from this picture, show her some appreciation. She's earned it. He spent hundreds of hours looking through a microscope, painstakingly identifying the thousands of macroinvertebrates that we captured in our samples, and here are the results of her work. Extant literature describes EPT taxa richness of over 10 as being an indicator of excellent water quality. And as you can see here, even our most homogeneous site boasted a taxa richness of 11. I also used tolerance values developed by the Montana Department of Environmental Quality in 2012 to calculate the Montana-specific Hills and Hof's Biotic Index, or MTHBI. An MTHBI of less than 3 indicates macroinvertebrate communities dominated by pollution-intolerant taxa. And as you can see here, every site we sampled had an MTHBI of less than 1. This metric also indicates unlikely organic pollution. Now I want to draw your attention to these two critters, the Rocky Mountain tailed frog and the Northern Rocky Mountains refugium caddisfly, both of which we collected in Sheep Creek. The Rocky Mountain tailed frog is listed as a potential species of concern by Montana. Numerous studies document local extirpations due to increased sedimentation and water temperature following road building for natural resource extraction. This caddisfly is a species of concern to the U.S. Forest Service, ranked globally rare by NatureServe, and is listed as a potential species of concern by Montana, with habitat degradation and mismanagement among the largest threats to species persistence. Both stand to be severely impacted by mining activities. Federally protected bull trout are extremely thermally sensitive and rely on clean, cold mountain streams. This, in combination with their cultural significance to Salish Kalispé peoples, led me to center this portion of my research on them. To understand bull trout distributions in the watershed, I conducted spatial analyses on data collected by the Forest Service. I found that nearly half of documented bull trout distributions in the watershed are downstream of the proposed mine. As such, mining the Sheep Creek area could reduce occupied bull trout habitat by nearly 50%. Similarly, rare carnivores face significant habitat loss if the mine moves forward. Whereas less than 8% of landmass in Montana is suitable habitat for federally protected Canada lynx, over 70% of the watershed is suitable. The watershed also serves as a connectivity corridor for federally protected grizzly bears, connecting the greater Yellowstone ecosystem to the Bitterroot Ecosystem Recovery Zone. The figure on the left from CELS 2023 shows predicted female grizzly corridors, with the yellow circle denoting the watershed where my research took place. The watershed is likely to serve as a dispersal corridor for female grizzlies, with the Hughes Creek area having the highest potential. Managers have postulated that this area will serve a key role in grizzly recovery. Okay, I just threw a lot of information at you, but what does it all mean? A few things. The Woods Creek West Fork Bitterroot River watershed has high quality water teeming with pollution intolerant macroinvertebrates and federally protected fish. The watershed also provides excellent rare carnivore habitat and plays a key role in dispersal for grizzly bears. Now that we all have some understanding of just how ecologically significant the watershed is, I'd like to share some recent updates regarding the mine. In April 2024, the Montana Environmental Quality Council petitioned Congress to mine near Butte instead. In November 2025, the mine was added to the FAST 41 list, which fast-tracks the project despite intense public backlash. In December 2025, Ravalli County Commissioners voted to unanimously write Governor Gianforte, urging him to support their goal of removing the mine from that list. By November 2026, the Fish and Wildlife Service will publish an environmental impact statement and record of decision for the Bitterroot Ecosystem Recovery Zone, Grizzly Recovery Zone, which may influence mining activities there. Based on the data I've shared here, I make the following recommendations. The Sheep Creek mine should be removed from the FAST 41 list immediately. Occupancy and predictive path level analyses of Canada lynx should be conducted prior to any mining activities. The grizzly bear EIS and ROD should be reviewed immediately upon release. Additionally, meaningful consultation with the Confederated Salish and Kootenai tribes must occur. This watershed is the heart of Aboriginal Salish Kalispé territory, and tribal members today remain reliant on the area for the continuation of traditional activities. Above all else, these data make a compelling argument for abandoning plans to mine in the Bitterroot and focusing those efforts on surface mining elsewhere in the state. Similar quantities of rare earths have been identified near Butte, and rare earth mining efforts should center there, not at the headwaters of a world-class trout fishery. And with that, I'll take any questions.
Any questions from council?
Yeah.
We were just talking about this morning, so it's timely. Any questions from the audience?
Say she just devastated us. I mean, I'm just totally floored by even this consideration, but all of the things that are being shown, it's really hard to watch.
We were learning a few new things from some studies coming out on selenium up in the Elk River Valley and the wildlife populations up there, on, specifically on birds that we didn't know about. So anyway, just the selenium ones, sometimes it gets missed. And I'm glad you included it in your study.
Yeah, absolutely. Selenium will play an important role in whatever monitoring critical materials decides to do following, if they do implement mining activities there. Selenium can lead to underestimating mercury toxicity in the long run.
See any more questions? Matea, you? Nothing?
All right. Thank you, Megan.
Thank you.
Great project.
All right, we'll move to our next presenter, Kobe Gierke. He's the Executive Director for Flathead Lakers. And here he is. Hello.
Hello, council members. It's a pleasure to be back here again to tell you a little bit more about the Flathead Lakers and what we're up to. So those of you who don't remember me from this last winter when I was in seeking a support letter for a project, my name is Kobe Gierke. I'm the Executive Director of the Flathead Lakers. I know most of you, but for those of you who I don't know personally, nice to see you again. So with that, I wanted to tell you a little bit about the water quality sampling work that we've been doing around Flathead Lake for the last 8 years. And if we have time, I'll tell you a bit about an invasive aquatic invasive species prevention project we're working on. So, quick background on the Flathead Lakers. Since 1958, we've been working to protect water quality, ecosystem health, and a high quality of life in the Flathead watershed. This particular project is something that we started in partnership with an open water swimming group back 8 years ago. And the open water swimmers were concerned about getting sick, swimming in waters where bacteria levels were too high for safe human recreation. So we started doing this sampling, and we've continued to do it because A, we think it's important, and B, it helps us identify trends in water quality and identify ways that we may be able to address that. So what we do is we collect samples from 16 popular access sites around Flathead Lake. Some of the ones here that you guys are familiar with: Salish Point, Riverside Park, Wolf Point, Blue Bay, Finley Point, just to name a few. And we work with the Flathead Lake Biological Station to have those samples analyzed for E. coli bacteria. Once we have those results back, which usually come back in just a couple days, we post that information on a website called swimguide.org. And that is a popular website used by swimmers all over North America to make sure that the sites they're planning their swims at are indeed safe to use. So when we have tests that exceed safe thresholds for E. coli bacteria, we'll also share that information on our social media channels and website. And then at the end of all of the year, we report all of this information to FWP, Flathead Lake Biological Station, and the Montana DEQ. This year, again, we tested 16 beaches, and for the most part, the lake remained very clean this summer. We had actually no tests exceed the federal threshold of 100 or 235 colony forming units per 100 milliliters, and just a few days or a few weeks where we had spikes at Bocher Park and Salish Point. So that's a pretty good summer for this. Over the last 8 years, those two parks in particular, Salish Point, Riverside, and Bocher, all have tested over the safe levels for E. coli bacteria multiple times throughout the summer months. So we were delighted to see that we didn't have any long sustained spikes of bacterial infection in the waters this year. That all helps us make a big important decision, which is what we were here in February asking for a support letter for, which is a stormwater collection project at the Polson Bay Golf Course to collect stormwater running off the golf course and allow it to work through natural filtration processes before it's released into the lake. So in 2026, we hope to begin that project and actually start digging in March. And this work that we've done over the last 8 years, that we've done with the sampling collection permits that we've received from you, have allowed this bigger project to take place. So I'll talk real quickly about the AIS project that we're working on. This was born of a partnership that we've long had with Fish, Wildlife, and Parks and the CSKT Natural Resource Division program to prevent aquatic invasive species. So we believe that if and when there's a potential infestation of aquatic invasive species, particularly mussels, it's going to come from outside of this watershed, and it's going to come out from outside of Montana. So we have asked FWP to share with us their data from their visitors. So anybody that made a campsite reservation at an aquatic-based park in Montana and also bought an AIS prevention pass from FWP twice in the last 6 years, we have their information and know who they are. And so we can start reaching out to those people while they're planning their trips and encourage them to consider the impact that they might have if they're not compliant with mandatory inspection requirements in Montana. So the way that we're going to do that is we're going to analyze this data that we've collected of 21,004 of individuals from locations of known infestation that have made multiple trips to Montana in the last couple of years. And we're going to reach out to them where they're at via social media, email, direct mail, radio and TV ads. And then we're going to provide incentives to help them make good decisions to keep the watershed clean. And then, of course, we'll report on our findings and the success of this to partners like FWP, like CSKT's Natural Resource Division, and others. So here's an example of what we're talking about. This is when I say high-risk individuals. This is western Minnesota, and you can see all those green and yellow dots are quagga or zebra mussel infestations. So we're looking at a 50-mile radius of those. Those are folks that are living in those communities that have been coming to Montana multiple times the last couple of years. That's who we want to talk to. Here's another example of that in the Las Vegas region. So our timeline is we started last April, and then our outreach campaign will resume in April of this year, hopefully prior to MAC Day starting. And then by autumn of 2026, we'll be reporting and working with our partners on that. So one question that I had is, where are these people primarily coming from? The two top states for folks coming that are in this target subset are from Minnesota and Texas. And these are people dragging boats to Montana and coming here and spending time on our waterways. And that's really the risk that we face trying to prevent these from being accidentally introduced. So we hope that this is going to increase awareness amongst folks that are coming to the watershed and in compliance. And of course, we really hope that this helps collaboration between FWP, between CSKT, and the other partners across the state working to prevent accidental introduction of invasive mussels. So that's my presentation. I'm more than happy to take questions.
Martin?
Thank you for, coming in and your partnership with the tribes and the different organizations out there for the aquatic invasive species. I've had the pleasure of being on those since I've been on council, and really important work. And so thank you for helping us protect our lakes. Much.
Any questions out there in the audience? Oh, Mr. Stark.
Yep.
Once folks get this problem, have idea what to do to get the problem?
Great question. We've actually got a couple of recent examples of that. The Snake River near Twin Falls, Idaho had mussel veliger detections starting 2 summers ago, and the same thing happened at Highline Reservoir in Colorado near Grand Junction. Both of those infestations, they didn't find adult mussels to start, and so they felt that they could effectively treat the veliger infestations, the microscopic plankton-like state of the mussels, with a copper-based solution, and ended up really, especially, well, in both situations, they had a lot of bykill of fish and other aquatic species as they tried to introduce this copper as a control agent of the mussels, to very limited effect. Both infestation sites in Colorado and Idaho are still testing positive for mussel veligers under microscopy. That's a tough word for me.
Go ahead, Martin.
The tribe has been working with FWP, with Tom Wolf, who's the invasive species coordinator for the state, on an incident response. And so they've actually had a couple trainings here in Polson over the last few years. And so we know that it's not if, it's just when they get here. And so education is probably the best thing. The clean, drain, dry, our boat check stations are really important. But again, we got to be prepared for when it does get here. So yeah, thanks for the question.
Yeah, I would like to add that CSKT has been one of the most active partners across the state in first identifying this threat and then actively working to prevent it. You guys are the only folks that run a 24-hour all summer long check station down at Ravalli that I think prevents a lot of accidental introduction. And in fact, we have a little ways to go. Flathead County Conservation District had volunteers stationed at the Summers fishing access site during the weekends in June, July, August, and they counted 124 boats launching from that site that did not meet mandatory inspection requirements prior to launching, meaning those people came from outside the watershed, launched anyway with no inspection. So it's one site, just weekends, just in the summer. There's obviously a lot more missed.
Yeah, it's a very difficult thing to overcome. It was, we did some training ones where we might have an outbreak in Summers Bay or, something like that, and trying to deal with it. You're not getting rid of it once you get it. It's just a matter of trying to keep the rest of your water bodies nearby from also getting it. And there's been some strong programs out there in other states like Minnesota where they've been very successful in containing it once they have it. But a lot of people just don't understand the risk until they finally see it. It's in their face. It's in their backyard. And then that's when they realize they need to do something. They need to be aware of their footprint and what they're doing, what they're transmitting. And so it's kind of like the Missouri folks, the show me state. You got to see it with your own eyes. Otherwise, you're not going to really pay attention. So the redundant systems that we have here in Montana have been worked out really good. Flathead Lakers have been a great, strong advocate for maintaining our mussel-free state, and particularly the Flathead watershed. And to have a redundant system where we're checking them on the borders of Montana, then we're checking them again on the Continental Divide, western Montana, and then we're checking them a third time for the Flathead watershed. There's a fourth inspection process, I guess two, one for Whitefish Lake and one for Glacier Park. And the best program is the Glacier Park program. And that's where basically you don't bring a boat in there and launch it unless it's sat through a quarantine system. You got to use existing boats that are there. And that's your best preventative tool. So hopefully we'll be okay. I tell this story from the summer where a famous person that lives, actually has a house on Lake Coeur d'Alene, but his boat was being transferred from Lake Havasu, I believe, up to Whitefish Lake, and it had mussels on it and was caught in Ravalli. So the worst thing about it is that this person knows better. Wayne Gretzky knows better than this. But how did that boat make it to Ravalli? It had to go through multiple check stations. And the worst thing is that those water resource managers of an affected water body allow boaters to go on that water body and boat and then don't quarantine them or clean that boat before they're allowed to leave. That's inappropriate, irresponsible, and a tremendous liability for the rest of us. So hopefully in a program in Montana, once we do, we know that we had it over there on Lake Elson, the reservoir, Tabor. I always forget that. Tabor. That was great. That was a great project. Great that it didn't turn out to be a real population that had started. But what a wake-up call for us, because it allowed us to go, oh my gosh, we got it here. We got it right here in the upper Missouri, and it's all these people are moving around. These anglers are moving all over the place. And how do we contain this? How do we stop it? And that really was a great wake-up call. So whoever went over there and dumped in those villager parts into that reservoir and we picked it up was, or actually a hero in my mind. But that really woke us up. And that's what it takes. It takes that immediate threat to say, this is what I have to lose. And Flathead Lake is in all the water bodies in our Aboriginal territories are very important for us. So all the work that Flathead Lakers do are fantastic. Great partner. James?
Yeah, the Canadians would have a problem with Wayne Gretzky and his selection of friends showing up on the golf course with the illustrious president. So I know there's a lot of people that are. Tom had an opportunity to get Wayne Gretzky's autograph, but we'll let him tell you that story off a different time. So, but thank you so much for your work at the Flathead Lakers. Thank you so much for the presentation.
Pleasure to be here. Thank you for having me.
Thank you, Kobe. All right. Thank you everyone for hanging in there. We have two more presentations. So we have Dr. Denver Holt, the founder and president of the OWL Research Institute.
But Denver, you need to use your press the button on your speaker there. Just press it once and the red light will come on.
Okay. Okay, cool. Thank you. Yeah, first of all, thank you very much for giving me the opportunity to talk. It's been a long time since I've been to tribal council, and recently I've talked to the cultural committees about some of the work. I've been doing work on the res for, 35 years now. And so to sum it up in 10 minutes is going to be pretty difficult. I'm going to highlight a few of the projects that we've have going here. But just to give you an idea, I first started doing work up here when you hired Dale Becker as your wildlife manager way back when. And I'm still going, and many of the other young tribal members who have become the biologists first started their wildlife careers with me doing research on the res here with owls. And it goes farther back to Billy Swaney, you said, who I had as a young student, and then from Billy to Stacy Corville and Dan Lipscomb and Shannon Claremont and on up to Carrie and Whisper and Peyton today. So I've been around a long time up here, and I'm just going to kind of give you an idea. All right, how do I make this go forward? Just, oh, this way here? Okay, cool. All right. So anyway, that was it. So just to give you an idea of some of the projects we have going, long-ear owls, a lot of it is between Missoula and the res here. So you'll see some accumulation of information from that. And some of the things that we're looking at is, why so many owls come together like this, if you can see it in the wintertime. And are they the same birds, and are they coming back to the same roost sites? And so we do a lot of mark recapture in order to figure that out, and a lot of habitat evaluation. And this is for the long-eared owl study. And then, of course, we have to follow them through the breeding season for the same things, looking at the habitat, what are they eating? Are they returning to the same areas to roost in the habitats that they utilize? And just to give you an idea, Pablo's also included in this, and to give you really what I think is you just can't study things for 1 or 2 or 3 or 4 or 5 years. You really have to put an effort into it, because you're going to see highs and lows in your populations of most wildlife species. So the Owl Institute over the years has always, kind of been a proponent of long-term research. And this just gives you an idea. There's up and downs. And so if you were to study them during a certain period of time, you would conclude this. Another period of time, you would conclude that. But over a long period of time, you would see that there's trends and there's up and downs. Another one of our projects in the grasslands here on the res is our short-eared owls. And this is the short-eared owls that we'll see on all kinds of properties out here, federal, state, tribal, et cetera. Now, if you look back in the early 80s or late 80s, early 90s, this is when Dan Lipscomb worked in this project. We had a high number of nests, and they were very successful. Then there was a break in the action, and if you look from about 2014 to present, we don't have a lot of nests, and they're very unsuccessful. And so we found that predation seems to be a problem, but we weren't sure what it was. We kind of thought it was just raccoons, maybe foxes from time to time, and skunks, more skunks than raccoons, excuse me. And so anyway, we tried to figure that out. We set up some camera systems in the valley and some of the conservation lands. And what we found out, surprising to us, was that coyotes were a huge problem. And not only were they eating shorter owl nests, they're probably eating all the ground nesting species nests. And we had never really considered coyotes until Beth set this up. Some of the other things that we're looking at here on the res are the small cavity nesting owls. And so these are the pygmy owls, and we're trying to find out what are their nest site characteristics. Are they reproducing? Are they successful? And are they returning to the same areas and their habitat requirements? All right, another species here. These small cavity nesting owls are very, very easily overlooked. And so we're doing the same things, all right? What are their nesting requirements? What are their habitat requirements? And are they reproducing? Are the same birds returning to the areas? And are we doing enough as far as saving their nest sites for the future? And I'll get to that. Same thing here. One of the things that Dale first had us do way back was go up into the mountains up in Chamberlain and the upper missions here and do surveys on snowmobiles. It was kind of tough to do, looking for boreals, and we found the first records of boreal owls nesting in the high elevations in the Mission Mountains on the res here as well. So same things. We put a series of nest boxes up because it was so difficult to find them, and we try to monitor how their populations are doing. Are they returning to the areas or not? And look at the general habitat stuff. So this is just, some of the basic biology that goes along with it is measuring, looking at growth rates and things of that sort, getting them all marked. And our migration studies kind of accomplish that. Our great gray one, this is a pretty popular one here. It's something that when we went to tribal council, cultural committee last year to get permission to put live cameras on the great gray owls because it's such a big and a well-known kind of an awesome bird. It's about this tall here with the wingspan of bigger, 5 feet or so. So what we're doing here is we're trying to look at, are the birds returning to the areas? How are they doing by getting the mark? What are the habitat requirements? And if you look at this big snag here, we're trying to get measurement data so that we can tell people, what are the requirements? Do we need to save some of these big broken top trees like that? So these are the types of things. Here's another again, another picture of a broken top snag, get all the birds marked. And just recently we have found some birds up at the base of the missions that have been, one, electrocuted, and also 2 cotton barbed wire fences over in the Swan side of this project. Traditionally, what we do is we go out on 4-wheelers, ATVs, snowmobiles, whatever it took, and do callback surveys. And now we're using these automatic recording units. So it kind of complements the physical activity where we can put these on trees. we can get detection rates, and then we can go in and that'll help us find more birds. We're trying to make comparisons, using both, is one better than another, and so forth. The barred owl here, you guys probably all know about that. Barred owls have been probably in Montana since the 20s. They're definitely on the res now. We're starting to find more nests, and the controversy is how many tens of thousands of barred owls do we kill to save the spotted owls. But just to give you, some insight into that they're here in the res, we're trying to get the habitat characteristics of, how they're doing their nest site selection, all that. This is a really big project here, given that, the res has become well known. It's been known for many, many decades as a mecca for wintering birds of prey. So you get eagles, hawks, falcons, and owls that come here in the wintertime and concentrate mostly on voles. We just came back from a conference in Costa Rica where we presented some of the preliminary information, and all the raptor people from the United States commented they know no other area in western United States that has the highest densities of wintering birds of prey as the Mission Valley, primarily located on the res from the lake south to, let's say, Mission. All right, and so the first 5 years of this 10-year study, we've recorded over 16,000 raptors of 17 different species. And again, I want to emphasize this is just 5 years of data, and you just can't go out and study things for 1 to 3 years. You need multiple years of data to see the trends. And this is what it looks like here from our lows of 349. Oh, whatever is this column here, you can't see what's going on, but the highest that we've had, oh, thank you. 999 counted in one day. Our next survey is this Saturday, and we'll see how it goes. It's all related to small mammals and voles. Outside of the bald eagle, if you look at the rest of these species here, they tend to trend with the voles. So even though the hawks, eagles, falcons, et cetera, are pretty cool, it's the voles who kind of determine the system. And we've learned that in the Arctic with lemmings and snowy owls. When voles in the Mission Valley have a good year, everything has a good year. When voles have a bad year, everything has a bad year, because everything switches to eating birds, eggs, nestlings, et cetera, of other species. All right, this is the snags. It says snags on the top. You don't have to worry about this, but what I want to tell you here is most owl species do not build nests, and they rely on either other species like woodpeckers to create their nests, or natural holes in trees and all that stuff. And so what we're trying to do on the res and among other areas is identify the trees that are really important. So we do know that firewood cutting goes on. We know that people take snags down for safety reasons, for disease. There's all kinds of reasons to take snags down. We're trying to give you some reasons to leave some of the snags up and work in conjunction with the logging industry, whether it's private or commercial. The tree on the left with the W on it is deemed a wildlife tree, I guess. It's a pretty lousy wildlife tree. It's really only good for insects and a feeding tree. The ones on the right with the woodpecker holes are the ones that we're trying to get the word out, take the measurement data, and save for these small cavity nesting owls that will not nest anywhere but in these trees. And this just gives you an idea. Douglas fir tree with the flammulated owl in it. All right, cottonwood tree with the screech owl in it. Aspen tree with a pileated woodpecker hole and a screech owl in it. Pygmy owl in I think it was a woodpecker hole here and an aspen tree. All right, solid owl and another aspen tree here. All right, burnt owl hole and a cottonwood tree. All right, and so what we're trying to do on the res and off the res as well is to go out there, identify the trees, get some, some measurement data so that we can tell not only, foresters on the reservation, foresters throughout, United States, Montana, et cetera, in the world, how important these dead and decaying trees are for small cavity nesting owls. Without these trees, they can't nest. All right, and even broken top trees like this, so we would try to encourage, if you've got one on your property, you've got one somewhere, and a logging operation, if it's possible to leave it, and it's not a safety concern or whatever, then we'd like you to leave them, because a lot of, even the big owls here will nest on the broken top so they can't get in the small holes. And this is the great gray owl one here on the res. And you can see here, there's not a lot of these big trees that are broken top like that, but they're very important because these birds are so big that for them to nest in. All right, and even if you move along the reservoirs and along the riverways, we have cracks in the walls in here, and some things like the barn owls will go up in there, so we try to locate them and also get measurement data. But also, too, these old buildings are important for things like the barn owls, which aren't very common in Montana, but they are somewhat findable on the reservation here. Okay, some of the other things that we've been doing for years is just, getting the public involved in research, doing community events, and just trying to get the word out on a group of animals, these owls, that a lot of people don't know a lot about. This is our migration study, which goes on in the lake and also at the base of the missions, east of Nine Pipes, for example, where the community is allowed to come out on Friday nights and see us band and trap these migrating owls. We're starting to find now that some of these banded birds, banded in Canada or down in Idaho, are following the same routes along the east shore of the lake and going north and south. Also to try to get some public awareness of, baling twine. It's definitely an issue, and we've had lots of osprey in particular that pick up the stuff, and a lot of get entangled and a lot of die, both the adults and the young. You can see one of the dead ones hanging from a nest in the kind of bottom middle corner there. This is kind of a cool one too. This is, we presented to the two cultural committees last year to try to get permission to use our live cameras. Up in the right above the camera, you can see a young owl sitting up there, but these are the cameras that we use in order to provide access to the public to see what we do. And also we learn a lot because we're not out there screwing around with the owls. We can actually watch them and they don't know we're there. This gives you an idea. Now, if you look at the right panel there, those solar panels, this is the one we used this year, just up at the base of the missions. We trailed it in, had 1,000 feet of cable. We hit it the best we could. We put it on a great gray owl nest. And if you look at down in the left-hand corner, it says great gray owls, 260,000 views just from the reservation here, just to look at the great gray owls, over 800,000 views of our osprey nests, et cetera, et cetera. So it's a really good way to communicate with the public and to get coverage, not only of the reservation, but just, owl biology in general. And then, we, of course, we've been doing work for years and all that, so we write a lot of papers. That's me in the Arctic and all that stuff, so you don't have to worry about that stuff. And so I want to just kind of close this out with you guys. Hopefully this will work, so I'm going to tell you, you might have to help me here. I want you to see what these cameras can provide by not going out in the field and, research is disturbance. By going out there and just watching the cameras and we can see natural stuff that goes on. In this case here, the owl's sleeping. It's got a nest over to the right-hand side, but a predator comes in, a potential predator. And we're able to see these things now in real time that we could never see before because our presence, kind of indicated or dictated certain behaviors. So let's see if this works. All right, so it's sleeping. It detects the threat. The Cooper's hawk comes in. The owl's trying to get big. Cooper's hawks kill these guys and eat them. So it's getting big. I'm big. I'm bigger than you. I'm going to give a little posturing now. And when the threat goes away, watch what the owl relax. This is what these cameras allow us to do. This is a great gray in the missions this year. Here she is in her broken top nest, feeding her chicks. $260,000 people watch this year. And we all know when the snowy owls arrive in the Mission Valley in the wintertime on the res, they kind of like to hang up on Polson Hill. And when that occurs, not only here in western Montana, but throughout the United States, it's the biggest wildlife viewing event in the nation usually. Everyone, even if you don't like wildlife, everyone goes out to see the great white owl from the north. And so I want to show you, this is our project in Alaska. Not only do I work on the res here in western Montana, but in the summertime we work on the Inupiat Eskimo lands, and I live in Eskimo Village up there, and we study the snowy owls on their lands as well. And I just want you to show, this is kind of cool stuff. I just want you to see it because those owls that show up here in the wintertime are from Barrow, Alaska, and other places, and this is their nest. Females have their job, males have their job. It's a great relationship. Watch. And nest on the ground. We're having an eruption year across the country this year, and we are expecting snow owls to show up in the Mission Valley any day now. Thank you very much. Appreciate it. You can cut that off. Don't know if you have any questions, or if you do, great. If not, it's all right.
Any questions for Dr. Holt?
I don't have a PhD, so anyway, sorry about that.
I just want to say this, I appreciate all your work, working with Dale and you, it's been fantastic. And I know there's been some other noise out there and other disciplines, our agencies, and we've always been proud to have you as a partner, Denver. So thank you for your work.
Yeah, no, we enjoy it. We're probably the most active owl research group in the world, and it's located right here in the Mission Valley. All right, thanks. Any questions out there?
Okay, question in the back.
Any what? Oh, public tours. The question was, do we have public tours for the snowy owls? Yeah, no, we don't. I mean, when they show up, then I'll show up there and we just do talks to the public and stuff like that. Like a few years ago when we had so many of them up on Polson Hill there, even the old folks' homes in Missoula were bussing people up, and we would give lectures. A lot of the schools were bringing people up, and we would give lectures and stuff like that. So we don't have organized tours other than I just show up and talk to everybody.
All right, thank you, Denver. We are on our last presentation with Dr. Maya Pedersen from the University of Montana and Dr. Samantha Morjo from our Tribal Health Department, Physical Therapy Department.
Good afternoon, everybody. As you guys know, I am Dr. Samantha Morjo, PT with Tribal Health, and also our Interim Rehabilitative Services Division Director.
And I'm Maya Pedersen. Oh, I'll try this. I'm Maya Pedersen. I'm an assistant professor at the University of Montana in the School of Public and Community Health Sciences.
Great. And we've been to you guys before, but just to update, our collaborative between Tribal Health and the University of Montana is our Growing Older, Staying Strong partnership. It does community-based participatory research here on the Flathead Reservation. So just a reminder, this is what our research team looks like. It was previously a smaller list, but we have expanded quite a bit in the last short period of time, few months really. We've gotten several more staff members trained, and we also now have community members trained. So you can see over here, I'm not sure if you can see the mouse moving. On the implementation team side, we now have a handful of community members who have gone through our fall prevention program, and then are now certified instructors. And so our tribal health team, our University of Montana team, on the research side of it, in the middle is our advisory panel. And then on that left side is the additional team members who do implementation of the program, but don't necessarily do a lot of the research side. So quite a large team these days. I'm going to hand it over to Maya to talk about the goals of the program.
Sure. So this project is a community-based participatory research project, and I'm kind of deviating from the slide for a second, so you won't see me reading along there, but we also we'll call that CBPR, so community-based participatory research. And that research really started with a partnership between University of Montana and Tribal Health back in 2018. And the original goal for that project, it started pretty small, but the overall goal for developing the collaborative was to use this community engaged approach to improve and increase health, quality of life, and longevity among Native American older adults and elders living on the Flathead Reservation. And we started this project via a series of 22 interviews with elders who had been recommended and were recruited to talk about physical activity and what type of movement they did and how physical activity related to their health and what type of access they had, what type of resources they used to stay physically active. And from those really deep, in-depth qualitative interviews, we built programs and we identified ways that those interviewees had said they'd want to increase physical activity. And we tried to build from existing resources within tribal health, as well as across the community. So from that overall goal, we created these 3 overarching aims, which are 1, to improve physical and psychosocial health through older adult focused health promotion programs and offerings. One thing we heard during those initial interviews was that there just weren't programs, physical activity programs that were specific to older adults. Like maybe they'd go to a fitness center, but it felt like it was something that wasn't for them. There were a lot of younger folks there, and there weren't programs specifically for older adults. So we've tried to have it be specific to that age group. The next would be to expand community capacity to promote health among older adults. So kind of getting back to that slide, really trying to get folks involved in getting certifications to deliver programs and build that capacity for delivery of those health promotion programs within the community, as well as build partnerships and collaborations. And then number 3, to strengthen systems and environments to support and sustain healthy activities and behaviors among older adults. And we were just here a few months ago, I think back in July, talking about making some environmental changes for pedestrian safety. And so that's an example, thinking about not only how can we offer more programs, but how can we also think about community mobility and community engagement, older adults kind of trying to walk and move around in communities. So we have quite a few programs that are ongoing through Tribal Health. We have Walk with Ease, which was an evidence-based program that now we've worked on adapting throughout the years, and we now call that Our Walking Journey. It's a group walking program, and I think we'll talk more about these at our next slide, but I'll just go through and list these. You might have heard some of these names before. The Stay Active and Independent for Life we also call that SAIL. That's a fall prevention program using strength, balance, and cardio, as well as some education to prevent falls. Tai Chi, which I know we have some Tai Chi folks in the room here, as well as some SAIL participants, or at least one SAIL. Oh no, he's gone. Oh, no, maybe some others. We have a Food for Health program now, which is a produce prescription nutrition program. Bingo Size, which we haven't started yet, as well as the Community Mobility and Pedestrian Safety Project, which really focuses on the environment and transportation. Okay, I'll pass it back to Sam here.
Okay, so, sorry. As Maya mentioned, so we do our CBPR work. We start at the top level, and then so we work through our advisory panel. We have them, community members have oversight of what we do. So we're regularly meeting with them, updating them on where we're at with programming. Our interdisciplinary research team, that team that was on the left side of that slide earlier, we were meeting weekly, we're now bi-weekly, just to better accommodate people's times. And then we also bring in advisors on specific topics when needed. And so then through that circle of steps on the side of the screen there, you see the way we do things is we implement a program, and then we run through it, and then we gather feedback from the participants. And then through that feedback, we make adaptations to the program, and then we implement again. And so we kind of go through that cycle. And those programs that Maya was talking about, the cool thing is that we're in different stages of this cycle with each of those programs. And so like with Our Walking Journey, it started at the implementation one. And then as Maya mentioned, we now call it our Walking Journey versus Walk with Ease because of the feedback that we had from community members. And so we've made adaptations to it based specifically on what our communities are telling us. So that is the cycle. I don't remember its fancy name. The iterative process? Yes. Iterative adaptation. The iterative adaptation process, if you want the technical term. And so here is just quick information as far as the funding sources. We do everything through grant funding with tribal health support. You can see we have one from Administration of Community Living. We have National Institute of Health. I think we have 2 National Institutes of Health. Sorry, this thing is in my way. And so, yeah, we seek outside sources to fund all the work that we do so that we can continue bringing these programs and the research on the programs that we're implementing into our communities without having a huge financial burden on our systems. Do you want to cover this slide? Nope. Okay. So this is, oh, sorry. The slide was new to me. I'm actually leading this project. So we've talked to you guys very briefly about the NARCH grant. We were recently awarded, well, SKC was awarded a 2-year planning grant from the NIH to study how to bring a Native American Research Center for Health here to CSKT. We're in the process of potentially moving that grant under tribal health, but Matea, Maya, and I are all on that project. And so our goal at the end of this 2-year planning period is to be able to submit an application to actually have a Native American Research Center for Health here on the Flathead Reservation. And so we have now gotten approvals from both the Salish Kalispel Culture Committee and the Kasanka Elder Committee to move forward with that project. That is it. Any questions?
Questions from council? Martin?
If you have an elder who's homebound, do you guys go to their homes at all?
No, in the sense that we don't have a staff member that goes there. With Tai Chi, that was another cool progress in our project, is that Tai Chi was delivered remotely. And so we provided tablets and hotspots so that that could go to the home. We did submit, I don't think we had it on a slide, a tip cap application with IHS. And so the goal with that one is to then start building better partnerships with our CHRs in the TCC division. And so that we can start getting more in the home kind of support for elders. And so then we would work on trying to build either our remote delivery of that or partner with the CHRs to train them how to do those programs in home. Not yet. Unfortunately, home health especially as a rehab world, home health is a lot of red tape to sort through. Any other question?
Anybody in the audience? Yep.
Can I talk this way? So we were just awarded the 2-year planning grant in August. So 2 years from now, we would be submitting the application for funding to actually bring the NARCH here.
Yeah, so we would submit the application in 2 years, and usually, hopefully, if it's funded the first round, it would be within 1 year. So that would be 3 years, ideally. And then that next round of funding for the center is a 5-year grant. Renewable, indefinitely. So many of the other tribes that have this grant it's been ongoing for 30 years, 15 to 30 years.
All right. Thank you all for your time.
Thank you.
Thank you, Sam and Maya. Thank you, council members. We are done for the day. Thank you for sitting through all these presentations. And yeah, if you have any questions or feedback, please let me know, and please help yourself to some snacks over there.
Thank you for this. It's actually really helpful to see the end product of all these things that we approve. So appreciate it.
Yeah, James, go ahead.
Yeah, thank you for the presentations. I may not have had questions, but it's interesting to see the research that's going on. And the other thing that's really important about what you do is making sure that research isn't happening without our input and our review. And so it's so important that, what you do and have